So, good morning and welcome to another Chai and Why. For the ones who are new to Chai and Why is CIFR's outreach forum, and it happens on first, third, and fifth Sunday of every month. On first Sunday, we are here at Prithvi Theatre, and on third Sunday, we are at Ruparel College. And if a month has a fifth Sunday, then we typically take you on a virtual visit to one of the laboratories of CIFR. So these days, we are actually discussing the work that was recognized with Nobel Prizes this year. Last session, we discussed the Nobel Award for Chemistry, and today we have Suchit. It's a pleasure to introduce Suchit, who is now become my friend over last couple of years since he joined TIFR. So Suchit is a research scholar in biological sciences. I may wish to say here that he is not a doctor, he is a biologist. And today he is going to discuss about the work that was recognized with this year's Nobel. So, thank you, Ullas. So yeah, first of all, I'd like to begin the talk by being extremely thankful to Arnab as well as Ullas for this opportunity to even talk about this. And second of all, it's really exciting to talk about this particular Nobel Prize because many of us were very keenly watching this time, and we were very eager to see what discovery would be getting a Nobel Prize. And when we heard it was this, we were extremely excited. And the reason for the excitement was not just the fact about the kind of impact it has had with the pandemic, but the kind of potential it holds. And today, what I would like to talk about is basically the discoveries that went behind, making this entire technology possible. So just before I start, what do you guys think the talk is going to be about? Like, there is a very weird title here. If you have studied biology, there are a few things you can guess based on what the title is. And if you're not, then it's going to look absolutely absurd, at least the first sentence. So what do you guys think the talk is going to be about? You can throw single words as well. Yes. Vaccine for COVID. Yes, that's the keyword. That's one. Anything else? Yes. Gene therapy using mRNA. That's in fact a very interesting answer because that is not something that we directly talk about when we talk about mRNA related vaccines or anything. Any other words, keywords, anything you think I'll be talking about? The first thing I thought someone will throw is vaccines. Like today I'm going to talk about how vaccines work. Actually, I'll introduce that. Yeah, you're talking about the mRNA, how mRNA is prepared inside the body, how you can prepare outside the body, how it can be put inside the body. One more sentence and you'll give my entire talk away now. So that's how close you were, and you will know exactly why it was so close. So to start with, again, a few more questions. It hasn't? Okay, let me just give away the questions also now then. Yeah. Must be fine on Zoom now, right? Yeah. Just try moving this. Good. Okay. Yeah, so let's start with a few questions, not about mRNA. Yeah, so has any of you over here ever been affected by chickenpox, or do you know someone who has been affected by chickenpox? You could just raise your hands if that's a yes. So that's like most of you guys sitting over here. And the next question. So did you or the person who was affected by chickenpox ever get it again? Like, was it a once in a lifetime thing, or was it once in a lifetime thing, right? Once in a lifetime thing.
And that's my question. Why do you think we did not get it again? Right. Right. So that the first word that you used, information, that is new, or so yeah. So it's somehow yeah. So that awareness or new, how did it get to know? That is what we'll be talking about, and that's the fourth question, which is there a way to develop immunity against the disease without having to go through it? Because this is an obvious question one would ask when you realize that, like, see, with chickenpox, while it's not as deadly a disease as if you were to compare this to another one, which is called a smallpox. With smallpox, it so happens that the mortality rate was high, which means the number of people used to die because smallpox was extremely high. It used to be 3 in 10 and whatnot. And that's a huge number. And in such a scenario, at least the survivors, the observation was that the ones who survived, you wouldn't see repetition of smallpox. What you would also not see is that um, that the survivors did not lead that great a life. What I mean, again, with respect to that, is smallpox was extremely debilitating. Sometimes, depending on where the pustules would form, it could even cause blindness, and that had major impact on lives. And smallpox, something, and that's an entire talk by itself as to how smallpox actually spread across the world. And smallpox is something that people across the world suffered from. So then, across the world, people wanted to find solutions in their own places, right? And the question is, how did they try finding solutions? So I think anyone who studied biology over here, if I were to ask this question now, which is, where did the idea of vaccination come from? Whose name pops up or what story pops up? Jenner. So Jenner is the first name that pops up. However, actually the answer links to something that I was talking about right now, which is that the problem regarding smallpox predates centuries before Jenner came into being. And you could actually trace it back to places like China, where smallpox was a major problem. And what they did, in fact, which was a traditional therapy of sorts, is what they used to do is they used to take these dried smallpox scabs. Anyone who's got chickenpox knows that they form these scab-like structures, like these pustules which dry up eventually. So the dried ones are blown into the nose of someone who's not infected. And what they used to observe is that whoever had undergone this therapy would not get smallpox again. So in a way, it was interesting that this idea of vaccination actually goes many, many, many years back from the history that we have been commonly reading about. But is it just China? It also goes to India, where something similar was also done in our country back then. And while the recordings come from, the recorded history from 18th century, there are reports which say that this could have been occurring for many years. Africa too. And Africa becomes extremely interesting from the perspective that, in fact, one among the Africans was actually captured and who used to work in America. So his name is Onesmus. I really must have gotten the name wrong. So he taught this method to cotton method. Like basically, this method is called as variolation. Like this is basically what vaccination is like. You take these in some or the other form. You expose someone who hasn't had the infection with this pustules, these dried pustules in some or the other form, and they end up developing immunity of sorts. And this method was taught, and that is how it, in a way, got introduced to America as well. And it again goes back to Europe too. So this by itself, this history is pretty interesting because it predates to back then. But why is Jenner's contribution so interesting and important then? That actually comes from the idea that over here, everyone is actually exposed to the scab of the smallpox itself. So this still would cause certain infectious states. And there was still mortality observed. And if I'm not wrong, numbers go anywhere from 2 to 3% of people. So going from 30% to 2 to 3% is not bad, but 2 to 3% is still a big number. And that is where Jenner comes into picture, or his observation that people with, he noticed this in milkmaids. So milkmaids, he used to notice that they used to get a milder infection that resembles smallpox. It's again a viral infection, but it's called cowpox. That's what they used to develop. And he used to observe that the ones who developed cowpox never ended up developing smallpox. And that was an observation he wanted to put to test. So what he did with this kid over here is he basically exposed the kid to these pustules of cowpox and then re-exposed the kid after a day to smallpox and notice whether the kid would actually end up developing smallpox. It so happens that the kid doesn't develop smallpox even after repeated exposures. And now that was, path-breaking, because this did not cause any major problem to the kid, unlike the previous variolation method. And these trials were repeated in 20 plus people, and as a consequence, they noticed that this was successful in all of them. So that became pretty revolutionary, because unlike the previous case over here, not a lot of people were dying, in simple words. So a much more effective way. And this is where the word vaccination actually comes into picture. And the first part of the word vaccine, vaca, actually refers to a cow. So that is the entire story of vaccination. And this by itself is, yeah, you have a question?
Yeah,
I get your point. This was happening in the late 1700s. Yeah, so yeah, because this was a completely human observation, and they were trying to find they weren't done then. Animals back then. Huh? Yeah, on the skin. That's how you. That's how it started. Jenner's work was like that. And then came this report, which you can see. So this basically shows, this graph shows on over here on the number of countries that report these cases. And over time, the number of countries that end up having smallpox is actually going down. So once upon a time, a deadly disease is actually completely gone right now, because you don't hear. Smallpox is one of those diseases, rare diseases. I think barely we have one or two such diseases which we have eradicated completely. So vaccination is completely responsible for this. But what is the idea behind, vaccination? And to understand how they work, I'll just give a basic glimpse of how our immune system works. So this is, as you all, everyone know that there is this defense system in our body, the immune system, which does this job. And it starts with the attack. And I'll talk about a virus right now, just convenient to talk because COVID. So as we know, we have this virus over here, which has these spike proteins on top of it. So do remember the word spike proteins, because later when I talk about mRNA, I'll eventually be coming to this. And because spike protein is what is on the surface of this virus. So this virus enters into our body and we end up developing an infection. Meanwhile, as we are suffering from this disease and the body is constantly trying to fight, it's also trying to learn. And what is it doing while it's learning? It's finding extremely tricky ways to take this virus, and there are certain cells in our body. This over here is called as a dendritic cell because of all these branches that it seems to have. And these cells end up taking this virus, tearing it apart into tiny pieces and presenting it on its surface. And later telling these other cells in the body, called these T cells, that this is something new that I found that I haven't seen around. And the moment this T cell figures out that it's something new, that is where the response starts. And you learn how to retaliate. And you retaliate in many ways, where you have some cells that end up producing certain chemicals that can help fight this infection. And certain other things that they also produce are antibodies. So antibodies are things that can now bind to your virus as well. And what do you mean by bind to the virus? Like if an antibody were to bind to this spike protein right here, the ability of this virus to even infect our cell is gone, because these antibodies are basically like tiny keys that will end up going and blocking the place where the lock keyhole is in the lock. So nothing else can actually go and sort of open this lock right now. And that is how the the COVID, the coronavirus actually enters the cell. It uses these spike proteins. So what is a takeaway over here? Basically that our body can learn and somehow remember this information, like how you pointed out. And the idea that if you were to somehow, you can actually prevent this infection if you were to block this spike protein. Now that is important to remember, and you can block that using this response. So what is it that we want to do when we need a vaccine or something like that? We want to be able to learn without this attack. So we wouldn't want to experience the disease, because this disease, as we know, is extremely debilitating as well. So we wouldn't want the disease, but we want to learn and be able to retaliate when necessary. And for that, we need vaccines. And there are various versions of vaccines, and I'm sure if I were to just show these variants, you guys can name the companies that I've thought of manufactured as well. I'm not going to take a quiz, just telling. So the ways typically we have developed vaccines so far is, I'm again talking about viruses right now, where you take a virus and then you weaken it of sorts. What do I mean by weaken it? The best example I can give you is all the dogs and puppies that we see around, which look all cute and nice. They were once upon a time wolves, and we have domesticated them and selected for the, in a way, I wouldn't say weak, but at least they're not as wild and ferocious as the wolves, right? So that is basically how we weaken a virus in the lab, by repeatedly culturing these viruses in, let's say, in chicken eggs and various other sources. And we keep selecting for the viruses that are weaker as in, like, they don't give rise to a disease eventually, but they still have these proteins. So exactly, and that is where we'll be going to as well. In fact, it takes that long for every one of these methods I'll be talking about. Like, even to get a dead or a killed virus and then eventually be able to use this to develop vaccines also takes time. Another way we do, which in fact is an interesting way, is you insert a DNA fragment that actually codes for some of these viral proteins into another virus, which we know does not cause a disease, so that this virus can infect us, but in the process, it ends up giving us the DNA, which will help us fight the previous infection. But we know that the viruses that are used over here are not the ones that cause infections at all. So they basically act as vehicles that take your DNA into the body. Another way is to just expose it to the proteins, like certain like this protein that you see over here. What does it remind you of? The protein I told you to remember, spike protein. So that spike protein that I was talking about, so you can just expose the spike protein to the human body in the sense, and the spike protein by itself does nothing, right? So you can expose that, and then the body learns to develop a defense against the spike protein, which is what we need if we would want to fight this particular virus. That is a broad idea. But as you had just pointed some time back, it takes a long time to develop. Many of these technologies take a long time to develop for various reasons, starting from the methods used to develop these components itself, right to testing. Takes a long time. But how long do you think it takes? And now let's say I'll only talk about in terms of just developing this material. I'm not even talking about clinical trials and all of that. In the lab, just developing the the material to start our experiment. Few hours, days, years. What's missing? Actually, it sits somewhere between years and months, and roughly the idea is it takes a year or two to develop. That's what we have seen. But I'm only talking about developing this material, not talking about this entire period, because that goes around for sometimes. Polio vaccine took around 6 years to develop, and many of these other vaccines took 10, 15 years to develop. Depends on which one we are talking about. But what about the mRNA vaccine against COVID that Moderna developed? How long do you think it took for them to develop?
Because January was event, and within 15-20 days they got the vaccine ready for them.
Yes, and that is pretty true. Like, you both are right if we look at the question in different ways, which is to develop this substrate itself, it took around 2 months. And to eventually go through all the clinical trials, it took close to a year, around 9-10 months, if I'm not wrong. I should just check the numbers. Yeah, of course. So we do preclinical trials. So a lot of this validation. So when I said that you're developing the vaccine, you also sort of include toxicology and all the other studies that are relevant to, which is preclinically done before the drug hits the clinic. So that is what I was referring to. So what is this mRNA? We are here. Yeah, you have a question. That's the talk. And that is the technology. And that's why I really got excited when I heard that this thing got the Nobel. So what is this mRNA? Now, before I talk about mRNA, I'll have to introduce you guys to where this mRNA is really found. What is it? What does it do? And that is important because mRNA was not discovered to make vaccines or therapy. mRNA was a basic science discovery. It was, a consequence of trying to learn more about ourselves, what we are made up of, how do we work. And this is the time when molecular biology was at its boom. What I mean by molecular biology is biology involved mostly studying organisms. But now we are trying to understand at the level of individual cells, within these cells, what happens. So if anyone finds the term cell a little alien, cells are basically the bricks of our body, quite literally. Because if this is what a brick wall would look like, this is what cells of an onion peel would look like under the microscope when zoomed. So if you just take a peel of an onion, which looks like a sheet, if you were to zoom it, it's made of tiny bricks. What you're looking at are mammalian kidney epithelial cells. These, in simple words, these are some cells found in the kidney, and you can see they form these really nice tubes. Do you see brick-like structures there? And each of these brick-like structures are called as cells. And while I call them as brick, it's better to call them as a cell because the first person who observed these cells, Robert Hooke, he called it a cell because it looked like tiny rooms. And room is a better analogy because there actually is a lot of things kept inside this cell. And if you were to look at these cells, they don't come in a single shape or size. Just like if you had to build a house, it's not one material that would be enough. You would need various different kinds of things, whether you're building a window versus a wall. You have various kinds of cells in the body doing different types of functions. They are sometimes made of different materials as well. I'll talk about what those materials are in some time. You already know all of it, but yeah. And they're of different shapes, functions, everything. The question is, how do they know what proteins to make to get this function? And the reason I talk about proteins is because if you see what a cell is basically made up of, or what our body is made up of, it's basically these biomolecules over here. We call them as biomolecules because that's what our body is made up of. And they're nucleic acids, carbohydrates. At least some of these things you might be familiar with, right? Like carbohydrates and proteins, part of our diet. So the idea is whatever we eat, we digest it into tiny bits and we reassemble it into the material that we would like to use. And that is the whole idea. For example, if you see nucleic acid, when I'm talking about nucleic acid, this material over here, you will realize in some moments that it's not really a stranger, that this nucleic acid is also made of things like sugars and certain other chemicals. So these are all mix and match of various chemicals that we consume, and we make use of it in interesting ways. Why do you say interesting ways? Because when I refer to these as chemicals, they are not just things that are floating around, hanging around. They are actually, if you were to have the ability to look at them, they would look like extremely tiny machines. They are not, they look tiny, but what you're looking at right now is just at the membrane or membrane of a cell. This is an animation by this, but I showed you a cell sometime back, right? Like it just had a membrane. But if you had the ability to look at the membrane, you'd see these things. So you see proteins all stuck in the membrane, some outside, some inside, things floating around, moving all the time. And while they have different shapes, some proteins also look like this. You see something's rotating over here. You have, I mean, innumerable number of these machines out there in your body. This is called as an ATP synthase. It's again a protein-based molecule. It's an enzyme. It's an enzyme because it actually makes something called as ATP. You guys have heard of ATP? What's ATP? What does it do? Anything? Provides energy. Okay. So that is the broad answer. And first, when it was discovered, you used to draw ATP as a blob. But if you actually look at ATP right now, it's a fully functional machine that is churning out ATP as it's rotating. And you also have other proteins that sort of walk around like this inside the cell. And then this thing over here that you see, this is called as a microtubule. That's basically the highway on which things walk around inside the cell. But so those highways are constantly formed, broken. And what you're looking at right now, unlike all these other things which were animations, this one's not an animation. This one's an actual cell you're looking at live, where we have interesting ways to actually look at proteins, label them with fluorescent molecules. So you can actually see them, see these things form, break quickly. There are two different proteins you're trying to see over there. So your cell is in a constant state of motion where it's not just, you know, a bag of chemicals. It's pretty active. It's an entire machine out there. And it uses this machine to do processes like this. So this is a cell breaking apart into two. The reason I spent this much time talking about this is one of the key molecules that makes all of this possible is proteins. And these proteins are tiny machines. So it's been extremely interesting as always to understand how processes like this occur. Because we knew cell division occurs, but we never knew how they occurred. But we knew molecules, some molecules could play roles in this. And there were questions of this nature all the time. And one such question was understanding where do they get all their properties from? Like, what I mean by that is initially I showed you, right? Multiple cells, they look all different, which our body is made up of. How do they know what to become? And where does that information come from? That was shown to be coming from this blue dot over here, which is present in every single cell over here. And some of you guys might recognize it as the nucleus of the cell, in some sort a center, but a center for what? So people started doing initial biochemical characterization, as in to figure out what is the dot made up of. And then they found a bunch of components. They're like, okay, it's made of certain sugar, phosphates, it's made of these things called as adenine, thymine, guanine, cytosine. These particular chemicals are present in that. But the question was, if this thing is giving information to the cell to become something, how is it conveying that information? Like, how do we convey information? We write something and send it, right? How do these cells, you know, get to know this information? So that logic came about when the structure of DNA was eventually figured out by Watson and Crick, and not just them, a lot of people involved in that. Even Rosalind Franklin's data was important, and many such data together gave rise to this structure. Now, this structure was very exciting because this doesn't look like any other chemical, right? Something is lying around. This is interesting because you'll need to understand that whatever colors, the color code you see over here, like A, G, C, T, that color code actually maps onto that twisted ladder-like shape. What I mean by that is that red thing you see over there, that is actually a cytosine molecule that's been attached. So this is basically like a structure, a chain on which each of these, one of these four things are attached. Not in the same sequence though. So the sequence can be anything. And that is the entire idea. For example, if you had to try reading this with your own eyes, from this end to this end, what would you read it as? What would this be? You could just say ATGC as well, easier. This is ACCT. So just like how your eyes are able to read this, now, and I have just put a map over there for that entire region, what its sequence would be like. And there are two ladders, so there are, you know, such complementary sequences out there. But now the question becomes interesting. This, whatever thing that contains information is in the center. The proteins seem to be everywhere. How is information going from the center of this nucleus to this entire cell? Because the other clue was that these proteins are not made inside the nucleus. They're actually made in the cell. They're made in different places across the cell. So the question, somehow you'd have to be able to send the information from the nucleus to outside. And you need a messenger for that. And that messenger happened to be RNA. And RNA is basically the chemical nature of what that molecule is. But if you look at it, again, the logic is pretty interesting. It's made of a single strand, and it has these bases arranged. And instead of thymine, it has another molecule called uracil. In fact, they do look pretty similar, except for some chemical differences that you can make out right now. So RNA is made out of UCAG, and the logic is the following, where if you had, just a second, yeah. So this is RNA. That if you were, what do you tell? You're in your kitchen, you need to cook something, but you get a crazy idea that there is this one particular thing you want to cook, and the recipe is there only in that particular library in some corner. So you'd have to run there, somehow go to that library, find the book, write it down, get it out of the library, get it to your kitchen, and then start cooking, right? So that is the whole logic. And if a cell has to cook a protein, if it has to make a protein, it has to go through the same logic, where just like how your eyes were able to read the DNA, we have protein machines that can exactly read the DNA the same way and make this yellow strand that you're seeing, which is your mRNA. This mRNA, so what you're seeing over here is the nucleus right now, enlarged. Animation, everything's animation. You see, do you see through these pores, you have these strands jumping out? So let's just focus on one strand that comes out of this. Do you see it? It jumped out from here to here or here to here, something like that. And then this blue thing, what you're seeing is basically the factory or the kitchen where protein is manufactured. And it can read this mRNA and accordingly make a protein. Yeah, yeah.
Make RNA. It has a number of places which are defunct, which is not used in mRNA. How we can understand that I need only CCC for that?
Sorry, I didn't get your question. Could you repeat?
If a DNA has a long chain,
right.
DNA is creating RNA. For creating RNA, it creates simple RNA. It is not still an mRNA.
The interesting question is, where do you start reading from? Where do you stop reading? How much of an mRNA do you make?
Just sort of the person gone. That was not a
right.
How does the system understand that I need only this part?
And that is a brilliant question, because if I were to give a sentence to you right now and put a full stop there, you would consider that to be place where the sentence ends. Similarly, there is some logic with respect to how these,
it has
where to start as well, right? Introns. So that process is called as splicing, where you have some regions that are removed, mismatched, and exactly the way it understands where to start, where to stop. So there are sequences in our DNA which code for all of these things. And based on the sequences, what do sequences do? They basically inform these proteins whether to stop, start, or it recruits certain other proteins based on which you end up looping the DNA a particular way, and you do all of these different. Yeah.
RNA converted to mRNA, where you have a lot of codons used. Those are only specific codons, and so that those codons create the protein. Protein, not the other part which is already in DNA.
Right. Right. That process is referred to as splicing. And I think, so it's a slightly, I'll have to get into molecular details to just explain how it works. So for the sake of this presentation, I will go ahead, but we can definitely talk about it because that's a very interesting process. So, yeah, so you see this mRNA being formed inside, coming out of the cell, going there, and discovering this logic was mind-blowing back then. So this is the entire logic right now, that with DNA you can make RNA, with RNA you can make proteins, and proteins end up adopting these weird conformations that even it's basically like origami, but they're functional machines. So that is the entire idea, and these proteins give rise to the various traits that you see in a given cell, like if it's from a neuron to a muscle cell, all of these differences. You have, it's not just proteins that mediate it, of course, but proteins do play an extremely crucial role in the process. And now let's just go through this timeline from when mRNA was discovered, because it's in fact two independent groups that discovered in two interestingly different ways. And this by itself is a very interesting story as to how mRNA was discovered, how some groups actually theorized the idea that, you know, something like an mRNA could exist. How did they go about doing it? That by itself is an interesting story. And yeah, I can, if anyone wants to know more about it, we can talk and I can give you more stuff to read regarding that as well. So the mRNA is discovered 1961. But 1969, another interesting experiment is done, where what they now did is they actually took mRNA from a mouse. They actually they took red blood cells and then ruptured it open and took mRNA from it. But they treated it with a red blood cell mush that was made using rabbit red blood cells. Now, what they wanted to see is whether this mRNA from mouse would actually end up giving rise to some mouse proteins itself, because there are some differences in mouse and rabbit proteins. Interestingly, it did. So it showed that the RNA by itself, it doesn't matter the system it is put in, but the fact that the RNA is now in that system is enough to make is enough for the RNA to start making proteins. So it doesn't matter what the source is. This becomes extremely relevant for us, because what were we talking about? We are talking about mRNA vaccines. But where is that sequence coming from? It's coming from a virus. So this was one of this proof of principle experiments that showed that you could take RNA from one place, put it in another place, and it will work. So that was an amazing finding. The next finding came, which was even more interesting, was from 1984, a group from Harvard, and it's their team. They're quite a big team. So all of them were, they tried an interesting thing. They tried to synthesize this mRNA in a test tube, basically. And whenever we do anything, you know, outside the body in test tubes, in simple words, we call it as in vitro. And they found a way to synthesize this mRNA in a test tube in vitro. Now that was again extremely exciting for the reason that you don't have to rely on an organism. Why is this interesting? Because whenever we thought about vaccines so far, I said that to develop these vaccines for all of these components, you had to develop it inside chicken eggs or some biological source of some nature. Over here, mRNA is something that does not rely on that right now. You can actually develop it somewhere outside in a test tube as a chemical reaction. And that became interesting. Interesting to an extent that people sort of hit the idea that what if we try delivering mRNA into cell lines? What I mean by cell lines is there are ways in which we take cells from the body and actually culture it outside. So you can take cells from the brain, cells from the kidney, you can culture it in, and what I mean by culture is you grow it in a dish, basically, like a test tube, but a wider one. And you grow it in a dish, you give it all the nutrients it needs, and it keeps growing. So over there, they try to see if we can actually deliver this mRNA, because it's not really easy to deliver mRNA. Their important role was they developed the method to deliver it. So they found a lipid-based thing. Lipid is basically a kind of fat. It's kind of a fat molecule. So you sort of encapsulate this RNA in that and then deliver that lipid, because that's one way to deliver it into cells. And they were pretty successful with that. And this was a breakthrough. But what was also interesting is if you actually looked at Robert Malone's lab notebook, in which they make notes and, you know, of day-to-day records of all the experiments that they do. In one corner, he wrote something very interesting, that this mRNA could be used for therapy someday. Because his idea was that if this mRNA can be delivered into cell lines, it can be delivered into cells, it can be delivered anywhere, then in the future, this could be used as a therapeutic, and therapeutic in multiple different ways is what he theorized. So having the foresight that this has such a potential is a big deal. So that was extremely exciting to look at. So, and as he predicted, you could see studies started coming from the 1990s, several of them in fact, where they were trying mRNA in different contexts to see if we could develop some therapeutics. So they tried therapeutics against cancer, tried various such scenarios, but there was a problem because you can see that it was 10 years. Doesn't look like RNA takes really long to make. Doesn't look like the test might take that long. However, it was barely one or two studies coming in a few years, and they didn't really catch on, in simple words, because there were some roadblocks. What were the roadblocks? The roadblocks were that this mRNA used to actually get destroyed pretty soon, even before it could get into our cells. Like this is, I'm talking about studies where they worked with mice or olive, even cell lines. In fact, this mRNA did not make a lot of protein. So that was a problem because we needed to make lots of protein. That is the whole point. So some of the RNA is going in, it's making some protein, but not enough. And that is what they saw. This is not a lot of protein. I'll later show what I mean by a lot of protein. And the other thing was mRNA induces something called as an inflammatory response. So inflammatory response is also one among these fight back mechanisms that our body has. And these responses are associated with so the diseases that we go through with respect to infections. Over here, an inflammatory response is also responsible for some of the symptoms that you see. So that's not a good idea. And inflammatory responses can actually end up shredding your mRNA even more. Even before your mRNA ends up helping you develop your immunity, the mRNA is gone. It's not making enough protein to develop immunity. So now how do you fight that? And during this time, in fact, people faced a lot of trouble, in fact, getting into this. So many companies were trying to back out, and not a lot of people tried to continue in this field because mRNA was a difficult molecule to work with. It seemed very fragile, while the scope was huge. But solutions actually came from our two people who some of you guys might recognize by now, Dr. Katalin Karikó and Dr. Weissman. And they ended up doing some groundbreaking research. They were the ones who, did some of these super interesting discoveries that were responsible for them getting a Nobel, and also for this technology taking a leap in terms of what it could do. And all of this was actually interestingly catalyzed by a photocopier in UPenn. If some of you have, I saw you nod your head. I'm sure you know the story. The story is of how they actually met. So back then you couldn't open your phone, Google for a paper and just look at it. You would have to go to a photocopier and actually get copies of the paper and then read it. That's where those two actually met, and they struck up a conversation. You had this person here, an expert in synthesizing, making mRNA in vitro. You had this person over here, super interested in immunology, specifically these dendritic cells. So in the beginning, I spoke about dendritic cells, right? Dendritic cells are these cells that end up taking these proteins inside, foreign proteins, breaking it into bits, exhibiting on their surface, and then showing it around that this is foreign. Do something about it. That is dendritic cells, and he was very interested in those mechanisms, and those two got into discussions and tried to do things. However, it was not an easy road at all because of the kind of failures that were involved and the difficulty to even get funding to do research of this nature, because we are talking about a time when mRNA was considered a dead end. So it was extremely difficult irrespective of its state, and this on a personal note becomes a completely different story. And it was extremely difficult for them to do their research. But what was it that led to the breakthrough? And the breakthrough comes from this experiment over here. I'll try showing this pictorial representation to help explain the point. So you grow these dendritic cells in culture. Easier to do these experiments in culture. So what they did is they treated it with RNA, but RNA that is not a, that was synthesized in vitro in the test tube. And what they saw is that level of cytokines are high. What are cytokines? Cytokines are the chemicals that give rise to this inflammatory response that we talk about. This inflammation that we see in the body, that is a consequence of these chemicals that are released called as cytokines. Cyto basically refers to a cell. So it's the cells in the body that release this. And you saw high amount of cytokines. So basically that it is causing an inflammation. But what's an interesting observation they had is, now they tried treating these cells with different types of RNA. So one RNA became nuclear RNA, as in they just took mammalian cells, I mean cells that are its own in a way, and they took RNA from the nucleus. You don't see that strong an inflammatory response. That was interesting because RNA for decades was considered to be inflammatory. In fact, this goes back to I think 1963, if I'm not wrong. Since then till now, till 2005, in a sense, was considered inflammatory. Now that was a problem, and this was a very exciting result for them to look at. What was even more exciting is now they took cytoplasmic RNA, as I showed, like the part that is not the nucleus but still encapsulated by the cell is called as a cytoplasm. They took RNA from there. That also did not give rise to a strong inflammatory response. They took mRNA from the cell. That didn't have an inflammatory response as well. Like that was pretty interesting. So it looks like where this mRNA is coming from matters. If it's its own mRNA, you don't see that inflammatory response. But if the mRNA was made somewhere else, you see an inflammatory response. tRNA. So tRNA is another kind of RNA that is present in the cell. So cell has various kinds of RNA actually. mRNA is barely the tip of it. So tRNA is one RNA which it's actually involved in making proteins again. And we can talk more about all of these RNAs. But basically, the interesting pattern is that if the RNA was from the organism, it doesn't lead to an immune response, which makes sense, right? Because RNA is there in the cells. You wouldn't want your body to be in a state of inflammation all the time. You should be able to distinguish it. And it was important to figure out what this foreign mRNA is, because again, the logic now it all makes sense, because this mRNA in the test tube, it was actually synthesized using an enzyme called as an RNA polymerase, which was derived from a virus. So as a consequence, this mRNA that you are getting, in a sense, doesn't resemble the mRNA that we have. But what do we mean by resemble? I just sometime back told you what mRNA is made up of. So how are these guys any different from this? That is where, that is the question again they asked. And this was the second part of that super interesting discovery that they had, which was that, so do you see this weird shape over here? This is the same tRNA that we spoke about, and it's actually a single string like this. If you see, it traces around, right? It's a single string, like RNA is being single stranded molecules usually. You see the single string being traced around, but do you also see that it's not made of the standard A, C, G, and U? Do you also see some other letters in the mix, letters, symbols, whatever, different things over here? A question mark also, that also actually represents something. So what you're seeing right now is some of these components are slightly modified versions of A, G, C, and all of these nucleosides. They're called nucleosides basically. And in fact, if you see, such modifications exist everywhere. So if you were to, so this is a diagram that is just showing the various modifications. Each one of this is a kind of modified. For example, this is a modified form of uracil. This is a modified form of adenine, and what you're seeing is this various forms, and there seems to be only a little overlap between different groups. Like bacterial tRNA might contain these modifications, but only some of them might actually be in eukaryotes. Like mammals are eukaryotes, so it might so only some of them might be here. There is very little overlap, and this is one of the ways in which our body actually figures out whether the RNAs are or foreign. And that was the breakthrough discovery that made a difference, because the next experiment was the interesting one, where they were like, what if we take this RNA that was made in vitro, we modify it such that it looks like it's from our own body. And that's what they did. Pseudouridine. So like I told you about uridine, pseudouridine is a slightly modified version of uridine, which gets incorporated when this tRNA is made. So they found a way to make RNA with pseudouridine in it. And when they now tested it out, like initially you used to get this inflammatory response, right? And you used to see very less protein being formed. What they observed is with pseudouridine, you end up getting, both the problems are solved. You don't see an inflammatory response. You see more amount of protein being formed and being sent out. And this is important because now the body has a lot of protein to learn from and be able to fight against it. So if we were to now talk about our COVID vaccine, then at least the mRNA one, you develop an mRNA against one of these surface proteins, because eventually the antibodies that are developed can actually block this surface protein and prevent the virus from entering into the cell. That is the idea. And that is what they saw. So these guys found a way to make RNA stable, and the amount of protein formed increases, the number of days this protein, the mRNA sustains increases. So all these problems are like, it looks like it's magically solved. And it is these discoveries that paved the way. And the reason they even constantly persisted to do these experiments was because of what it offered. As in, they could basically see the future. Like many of these guys could see the future of what mRNA basically is. So our current vaccine consists, of course, of this mRNA with these modifications in the middle, and it's coated with these lipids. This by itself, a lot of research has gone into developing this, and that's a very interesting concept as well. So you coat it in this interesting way, and this forms something called as a lipid nanoparticle. This nanoparticle is sort of what is injected into our blood. It enters into our blood, and this gets taken up by those dendritic cells that I was talking about, which eventually end up taking this mRNA. They turn it into proteins. These proteins are digested and put all over, and then the body recognizes it, that's foreign, and develops an immune response without us having to go through a horrible disease. And that is basically what happens. This is just a representation of how it can enter the cell and then gets released. And it makes protein. So a timeline is what we were talking about in the beginning. Like, unlike the other methods of making vaccines, over here you don't need the sample. What is it that you need? You just need sequence. And that can be sent across a computer as well. Because based on the sequence, you can actually make the mRNA in the lab, you can synthesize it, and you can start working on it. So we can sort of be as fast as the disease in terms of developing cures. And that is what makes it so promising. And I'd like to end with the future potential. And I think the image is a bit tiny, so you might not be able to see all of it. But the entire idea behind these mRNA as a therapeutic is that the RNA can make proteins. That's the basic principle. So now you can make any protein. So that becomes important because that can be used to cure various kinds of diseases out there. And there are experiments at preclinical level going on right now, some even entering into clinical trials. And with respect to developing cancer immunotherapies, even with respect to certain vaccines against certain other infectious diseases, even with respect to allergy tolerization, you have these systems now being developed. And just to give you an example of what I mean by therapies that are completely against vaccines or cancer immunotherapy or anything like that is, you could actually do things like, in some diseases, there are certain proteins that are just present lesser in number in a particular cell of our body. So a method like this and these diseases are genetic, extremely difficult to cure, very rare to find. So developing vaccines or therapeutics for them becomes expensive and pretty difficult. However, with methods like this, it's extremely feasible. It's pretty feasible to develop therapies, making it so exciting. And the reason this entire field is exciting is because of what the future holds in terms of the various kinds of therapies that are coming in and more that are yet to come. So that's where I'd like to end my talk, but I'd like to I'd urge some of you to read this particular review, which is a tangled history of mRNA vaccines. Because whenever we talk about science, it's extremely tricky to nail down just one finding or just one person or two people or three people responsible for the whole field. This actually comes from micro steps that have occurred over decades, several years. And this is one such review which talks about the kind of contribution that various people have had, which if we wouldn't have, maybe we wouldn't even have had these vaccines. So that is the broad idea. And I'd like to leave you with that message. So thank you for your time and patiently listening. Did I take long?
So we'll take perhaps a few online questions. Break for tea. We'll come back and ask the speaker, the usual format. So there are questions like, was the sequence of the control mRNA different than the test sample? Perhaps if you can go back to the slide.
Which?
Before this. Yeah, here.
I think these experiments. Yeah, of course, the sequences were different, but it's not that this was a pretty homogeneous pool as well. It's not like it was just one sequence that was used. So in all of these, it's a pool of sequence. So you take RNA from the entire nucleus. So it's a lot of various sequences of RNAs, which is why they didn't think that it was a particular sequence that induced this state. And that's also because of these previous initial studies where they wouldn't use just a particular sequence of RNA. It was always an entire, a fraction that was taken. And yeah.
And the second question is about how does tRNA undertake loop conformation? The tRNA. How does it undertake?
And tRNA undertakes, and in fact, if you look at tRNA's actual configuration, it's not even as simple as this. This is how over here, we are only talking about how these two bases are forming bonds. But the actual loop, I wish I had an image to show that. The actual loop conformation it sort of looks like an L that is bent and it's a completely twisted structure. It looks like a mess. And that is the case with all of these biomolecules. And the kind of forces that usually play a role are things like hydrogen bonding. And as a consequence, you have this entire structure forming these various configurations. So depending on the sequence, you can actually have different conformations, even though this is a single strand. Any more questions?
Yeah. It's perhaps a bit strange, but why was the control mRNA used at all? Earlier.
Let me just go there. Oh no. Which control are we talking about over here? I mean,
why not use RNA?
oh, they took away. I'm showing 4 right now. If you actually look at their panel, I might have the graph. This was just a backup slide I'd kept for something. They in fact used various kinds of RNA over here, and you can actually see tRNA from E. coli ends up giving an immune response. So the entire idea behind the experiment was to just treat it with different types of RNA and see what the immune response is, to just understand that is this a general thing, because that's what they thought as well. Like if the RNA is from the cell, technically shouldn't cause an immune response. But why is RNA in general still, immunogenic? Why is it still leading to these strong inflammatory states? That was the whole question. So yeah.
What's the difference between the minus and plus here?
Minus and plus, these are actually different treatments. So what they did is, so this is a slightly nuanced experiment. What they did, they took RNA, and initially they wouldn't treat it with this chemical called, it's another protein called RNase. RNase basically breaks your RNA down into bits. So minus refers to, laser is not working. That's okay. Minus refers to when you don't treat it with RNA. So the RNA is intact, but plus refers to the time when you treat it with RNA, so the effect is gone. This is done to make sure that whatever effect you are seeing, this inflammatory effect, it's RNA that is responsible, because many of these extracts are from the cell. So there's a chance that you might have certain contaminants that could induce such a state as well, right? So to rule out that possibility, they treated it with things like RNAs, and then they don't see that response. So that is all. Yeah. Anything works?
Okay. Since it's already 12 o'clock, I'll announce the next session. So the next session will be 2 weeks from now at Ruparel College, and we'll be discussing about the work that was honored with Nobel Prize for Physics this year. Okay. So that will be 2 weeks from now. So now we'll take a small break. We'll come back and we can ask questions.
Yeah.
Okay. So thank you, Sujit.
Thank you. Thanks for the opportunity.
After tea, we'll come back.
Yeah, sure.